Hey everyone, and welcome back. Ready for another deep dive?
Definitely.
Awesome. Today we're going to be looking at the world of fluorescent proteins. Those glowing marvels of molecular
Oh,
biology.
yeah, they're really cool.
Yeah. And I know you're interested in figuring out how to choose the right one for your research, right?
I think that's something a lot of researchers struggle with.
For sure. So many options, it can be overwhelming.
Yeah.
But don't worry, we're going to help you navigate that whole process. We've got this fantastic guide from Assay Genie.
Oh, nice.
Yeah, it's called Choosing a Fluorescent Protein. A comprehensive guide.
Oh,
Very thorough, just what we like. And it's written by Tareem Ali, who has a master's in bioinformatics, so we're definitely in good hands.
sounds like it.
So fluorescent proteins, they let us actually visualize the invisible. It's incredible.
It really is. I mean, think about it. Actually seeing cellular processes unfold in real time.
I know, right? That's the kind of power we're talking about here.
Powerful stuff.
Okay, so I think you already know the basics of how fluorescence works.
Yeah.
They absorb light and emit it at a different wavelength, making them glow.
Right.
But with so many to choose from, how do you pick the right one for your specific needs? This guide breaks it all down into these key factors, almost like a checklist, which I love.
Checklist is great. Keep you on track.
Totally agree.
Hmm.
And each factor on this checklist plays a crucial role in whether or not an FP is going to be a good fit for your experiment.
Definitely. You really have to consider all of them.
Okay, so first up on this checklist, spectral properties. I know these have something to do with the wavelengths of light, but why are they so important? Can you break that down a bit?
Sure. So think about it like this. Imagine you're trying to listen to two different radio stations at the same time,
Okay, I can see where you're going with this.
but their frequencies overlap,
Yeah.
So what happens?
It would just be a jumbled mess of sound. You wouldn't be able to understand either station.
Exactly. And it's the same principle with fluorescent proteins.
Ah, I see. So it's about clarity.
Right. If you're using multiple FPs in an experiment, let's say for multicolor imaging, their excitation and emission spectra, they need to be distinct, meaning they shouldn't overlap too much.
Otherwise, you get that jumbled mess you were talking about.
Yeah, it's called signal crosstalk. Basically, the fluorescence from one protein interferes with the signal from another.
So basically, choosing FPs with well-separated spectra is like tuning your radio so you get clear, distinct signals, no interference.
Exactly. That way, you can see each target clearly and actually interpret your results accurately.
Makes sense. Okay, non-overlapping spectra check. What's next on our checklist?
Brightness and photostability.
Ah, okay. And those two kind of go hand in hand, right?
They do, yeah.
Yeah.
Brightness basically refers to how much light an FP emits.
Also, a brighter protein, stronger signal.
Exactly. Easier to detect, too, especially when you're dealing with complex biological environments.
That makes sense. A brighter signal is always a good thing. But what about photostability? What makes that so important?
Well, imagine you're trying to observe a cellular process over time, using a time-lapse experiment.
Okay, yeah.
If your fluorescent protein fades quickly when it's exposed to light, you're going to lose your signal before you can get any meaningful data.
Right, so you need it to last.
Exactly. So photostability is really all about how long it can hold up under the spotlight without fading away. Some FPs are like marathon runners. They can stay bright for a really long time.
And others are more like sprinters, bright but fade quickly.
Exactly. The choice depends on your experiment. If you're using high-intensity light or doing long-term imaging, you'll need an FP with strong photostability.
I like that analogy. Marathon runners versus sprinters. Okay, brightness and photostability, check and check. Now, this next one sounds kind of intimidating. Oligomeric state. Can you break this down for me?
It's not as complicated as it sounds. It really comes down to how the fluorescent protein molecules, how they arrange themselves. They can exist as single units, which are called monomers, or they can pair up dimers, or they can even group together in fours, which are tetramers.
So it's like building blocks.
Yeah.
Monomers are the individual blocks, and then you can have these different combinations like pairs or groups of four.
Exactly. And for most applications, especially when you're creating what are called fusion constructs, where you attach the FP to another protein, you'll want to stick with monomeric FPs.
Ah, okay. And why is that?
They're less likely to interfere with the normal function of the protein you're studying. If you use a dimeric or tetrameric FP, it might force the target protein into kind of an unnatural configuration.
So it could mess things up.
Yeah, potentially. Affect its behavior.
So monomers are generally the safer bet. But I'm guessing there are situations where those other forms might be useful.
Absolutely. Sometimes you might need those specific interactions between FD molecules, in which case dimers or tetramers might be necessary.
Right, because the research question demands it.
Exactly, and sometimes that extra brightness that dimers and tetramers can provide might be advantageous.
You just have to weigh the potential benefits against the risks. Okay, oligomeric state check, but with a little asterisk for those special cases. What's next on our checklist?
Maturation time.
Maturation time. Okay.
This refers to how long it takes for a newly synthesized FP to actually start fluorescing.
Oh, so some are faster than others.
Yeah, think of it like waiting for a photograph to develop.
Oh, so some FPs are like instant cameras, while others need time to develop their fluorescent.
Exactly, and that speed can be critical. If you're looking at very dynamic processes, things that happen quickly, you need an FP that matures quickly.
Right, otherwise you might miss something important.
Exactly. But if you're observing something more static, a slower maturation time might not be a problem.
Makes sense. You wouldn't want to miss a crucial event just because your FP is taking its sweet time to light up. Okay, so maturation time check. And last but not least on our checklist, cellular localization. What's that all about?
It's all about where in the cell your FP ends up.
Okay.
Some FPs are designed to target specific compartments, like the nucleus, the mitochondria, or the cell membrane.
It's like having a molecular postal code, directing the FP to its designated location.
You got it. And this targeted localization is incredibly powerful for understanding how different parts of the cell actually function.
So you can literally see where your protein of interest is located and how it moves around.
Exactly.
That's amazing. Okay, so we've covered a lot of ground here, from the basics of fluorescence to all those key factors to consider when you're choosing an FP. And it's clear that each of these factors really plays a crucial role in designing a successful experiment.
Absolutely. You can't just pick one at random. You really have to think it through.
Exactly. So now that we've got a good grasp of the fundamentals, are you ready to meet some of the most popular FPs and see how they measure up against these criteria?
Absolutely. Let's do it. I'm excited to see what we've got.
Me too. Let's meet those fluorescent all-stars.
Awesome. All right, so let's kick things off with the one that really started it all.
EOG.
Exactly, GFP. It's been lighting up research for decades now.
GFP, the OG fluorescent protein. I love it. It's really amazing how something that was discovered in a jellyfish ended up being such a game changer for science.
It really is. So GFP, or Green Fluorescent Protein, to be official, has a peak excitation wavelength around 488 nanometers.
Okay.
And it emits that classic green glow at around 509 nanometers.
Got it.
It's relatively bright, it's stable, and it's been extensively studied and optimized over the years.
So GFP, a solid all-around performer. But I noticed that the guide also mentions an enhanced version of GFP.
Oh, yeah.
What's the difference there?
Enhanced GFP, or eGFP, it's like GFP but with a boost.
Okay.
Scientists tweaked the structure a bit to make it even brighter.
Even brighter.
Yeah, and also more suitable for using in mammalian cells.
So brighter and mammalian cell friendly sounds like a winner.
It's pretty popular, yeah.
But I'm guessing sometimes you need more than just green. That's where those red fluorescent proteins come in, right? The RFPs.
Exactly. RFPs like M-cherry, they're often used alongside GFP, particularly for multicolor imaging.
Makes sense.
Remember that radio analogy we were talking about before?
Yeah, with the overlapping signals.
Exactly. So M-cherry, it excites and emits at longer wavelengths than GFP, so their signals don't overlap, meaning you can visualize two different targets at the same time.
So GFP and M-cherry, they're like the classic duo for multicolor imaging.
Yeah.
Perfect partners. But this guide goes even deeper. It even mentions far-red and near-infrared FPs. What's so special about these longer wavelengths?
Think of those as the deep-sea explorers of the fluorescent world.
Ooh, I like that.
These FPs, like M-plum and IRFP, they're particularly useful for deep tissue imaging because longer wavelengths, they can penetrate further into tissues with less scattering.
So they're like the submarines of the fluorescent protein world, able to reach depths that other wavelengths just can't. Love these visuals. But there are even some more exotic-sounding FPs mentioned in here, these photoactivatable and photo-switchable FPs. Those sound almost science fiction.
I know, they kind of have that futuristic appeal, don't they? But these FPs are very real, and they are incredibly powerful tools.
Okay, so tell me more. What are they all about?
Well, they can actually change their fluorescence in response to specific wavelengths of light.
Hold on, so we're talking about FPs that you can essentially switch on and off with light.
Exactly.
Wow, that's incredible. What kind of research can you do with that?
Well, imagine tracking the movement of individual molecules within a cell with crazy precision.
Okay, yeah, that's next level.
That's one of the possibilities these FPs open up.
Can you give us a specific example?
Sure, so there's PA-GFP, or photoactivatable GFP.
Okay.
It doesn't actually fluoresce until it's hit with a very specific wavelength of light.
And that activates it.
Yeah, it activates it, so researchers can then track only the activated molecules. And that can give insights into things like protein trafficking and diffusion.
So it's like having a molecular spotlight that you can aim to illuminate only the molecules you want to study. That is so cool. What about those photo-switchable FPs? How are those different?
So photoswitchable FPs, like Drampa, they can be switched back and forth between a fluorescent state and a non-fluorescent state, and you can do that multiple times.
So it's reversible?
Exactly. And this reversible switching, it's super useful for super resolution microscopy techniques because it lets researchers basically break the diffraction limit. You can visualize structures with incredible detail.
Wow, so it's like having a dimmer switch for your fluorescent signal, revealing finer and finer details with each switch. Amazing. It's incredible how each of these FPs has its own unique strengths, making it suitable for different types of experiments. But with so many options, how do researchers actually choose the right one for their needs? It seems like a tough decision.
Yeah, it's a great question, and it's something this guide addresses head on.
Okay, good.
Let's move on to the practical side, matching those fluorescent proteins to your research goals.
Okay, this is where it all comes together. I'm ready to see how all this fluorescent protein knowledge actually translates into real-world research.
All right, let's break it down then, starting with live cell imaging.
Live cell imaging. Okay, so for that, where you're essentially watching cellular processes unfold in real time, what are the top priorities when you're choosing an FP?
I mean, first off, you need an FP that can handle the spotlight, so to speak.
Okay.
Photostability is crucial there. You're continuously exposing those cells to light.
Right, so it can't fade too quickly.
Exactly.
Yeah.
You don't want your signal to fade away too fast. And of course, you have to choose an FP with the right cellular localization.
Of course, it's got to be in the right place.
If you want to study mitochondria, you need an FP that specifically targets mitochondria.
You need an FP that's up for the task and in the right location.
Hmm.
Makes sense. What about multicolor imaging? What are the key things to consider there when you're visualizing multiple proteins at once?
It really boils down to avoiding that signal crosstalk that we were talking about earlier.
Right, the jumbled mess.
You need FPs with non-overlapping emission spectra. GFP and M-Sherry, that's a classic starting point.
The classic duo.
But there are many other combinations out there depending on the wavelengths that you need.
So it's like assembling the perfect fluorescent palette
Exactly.
to paint that detailed picture of the cellular event. What about super resolution microscopy? How do those shape-shifting FPs fit into this type of imaging?
Super resolution microscopy really relies heavily on the unique properties of those photoactivatable and photoswitchable FPs.
Okay.
By precisely controlling their fluorescence, researchers can bypass the diffraction limit of light, revealing structures with, nanometer scale resolution.
It's like going from a blurry standard definition image to a crystal clear high definition one, uncovering details that were totally hidden before. What an incredible advancement. The guide also mentions a technique called FRET, First Resonance Energy Transfer. Could you explain how that works and how it relates to fluorescent proteins?
So FRET is a really powerful technique for studying interactions between molecules.
Okay.
It relies on this principle that when two fluorescent molecules are super close to each other, the energy from one can actually be transferred to the other.
Wow.
But this only works if their spectra overlap in a very specific way.
So they have to be compatible.
Exactly. You need to choose what's called a donor FP and an acceptor FP that are compatible.
So it's like a molecular dance.
Yes.
If they're close enough, they can transfer energy to each other, and we can see that transfer as a change in fluorescence. That's such a clever way to study protein interactions. This deep dive has been incredibly illuminating. I'm starting to feel like I could actually choose the right FP for an experiment now.
That's great to hear. And the field of fluorescent protein technology is constantly evolving. New and improved FPs are being developed all the time.
That's what makes science so fascinating. It's always pushing boundaries,
Exactly.
discovering new ways to explore the world around us. Speaking of pushing boundaries, the guide ends with a look at future directions in fluorescent protein development. What are some of the areas researchers are focusing on?
Well, they're always trying to develop FPs with enhanced properties, brighter, more stable, new colors. It's all on the table. They're also working on FPs that can actually withstand more extreme conditions,
Oh, well, like what?
like low oxygen environments or acidic pH levels.
Okay.
And that would open up whole new avenues for studying processes that were previously inaccessible.
So it's like they're building a Swiss Army knife of fluorescent tools,
Yes.
each one with its own specialized function.
It really is amazing, how far we've come with fluorescent protein technology, and the future is, well, even brighter, pun intended.
I see what you did there. So just to wrap things up, it's clear that choosing the right FP, it's crucial for any kind of fluorescence-based experiment,
Oh,
really.
yeah, for sure.
You need to think about all those factors we talked about. Spectral properties, brightness, photostability. Oh, and the oligomeric state, maturation time, cellular localization, all of it.
It's a lot to keep in mind.
It is, but you got to match it carefully to your research goals. It's all about finding the right tool for the job. And if you want to delve deeper into all of this, I highly recommend checking out the full article from Assay Genie, Choosing a Fluorescent Protein, a comprehensive guide, written by Tafreem Ali. It's a fantastic resource.
Definitely a great starting point for anyone working with FPs.
Absolutely. That was the basis for our whole deep dive today. Now, before we finish up, I want to leave you with a little something to think about. Given all the amazing advancements we're seeing with fluorescent protein technology, what other groundbreaking applications might be just around the corner? What other mysteries of life might these glowing proteins help us unravel?
That's a really interesting question. I mean, the possibilities are truly endless when you think about the power of actually being able to visualize the invisible.
Well, that about wraps up our deep dive into the fascinating world of fluorescent proteins. Hope you found it as illuminating as we did. Until next time, stay curious.